Hey, what's going on? It's Mr. Salmingo and Mr. Jackson. For this section for Decathlon, it's science. It's biology. So this short video, we're just talking about how do you make proteins from DNA. So you should already know that DNA is the code for everything about you. But how does it get from a code to actual stuff? And the actual stuff is proteins. We're talking about how do you get from DNA to the stuff that you are today. So only two steps we're talking about. Step one is called transcription. Transcription, the process of transcribing. I'm going to start here with step one. In step one, what you're doing is something called initiation, where you get this double helix, this DNA, right, Mr. Jackson? And we have this polymerase, this RNA polymerase. It's kind of like a Pac-Man, and it goes to the DNA and it sees what code. Remember, the code for DNA is like A, T, G, or C, and it finds the partner. So if there's an A, it actually has a U instead. RNA does not have a T. So if it sees an A, it creates a U. If it's a G, it creates a C. If it's a C, it creates a G. And it takes the DNA and makes its own strand based on the partner it sees in DNA. And that's what we're talking about elongation. We just keep making more, adding to the strand, whatever the complementary nucleotide is. And then once it's done making it, it'll see a DNA sequence that means to stop or termination. And it stops that RNA strand, and you have basically a chain that is a whole bunch of bases that is the complement of DNA. And the modifications that happen at the end of it, once I copy it all, I add what they call a poly A tail. It's a whole bunch of A's. So A, T, G, and C. It just adds a whole bunch of A's to the end, adds a tail. And then we talked about this today in class. We remove something called introns, so things that are not necessary or not needed for the proteins. They remove something called introns. They keep the exons for mRNA.
Cool.
That's what I thought. All right. The second step, again, there's only two steps we're learning today, is called translation. So we started with DNA. From DNA, we had the polymerase to make a chain of RNA. With that chain of RNA now, it's like, how do you make stuff? How do you make the proteins? Three steps again. The initiation. So from this mRNA, and I know this is kind of confusing because there's no pictures, but at the end of this video, we're going to show you an animation. So this chain of mRNA, a tRNA will come to the mRNA, and it has what they call a start codon. It's a 3-letter sequence that tells the tRNA to start on the mRNA. And that is called the P site. Okay. Next, this tRNA will bring an amino acid based on what letters it sees. So for example, if it sees an AGU, the tRNA will bring an amino acid that relates to, that is, I don't know what the word I'm looking for is, that relates to that code. If the code is CCC, then it's going to get an amino acid that corresponds to CCC. This happens at the A site. And again, it's hard to explain. There's no pictures, but I'll show you a video. And this A site is going to add the amino acids that are coming in. Okay? And then it ends when it goes towards that RNA sequence and it sees a codon, a 3-letter sequence that means stop, and it stops the sequence, and then this chain of amino acids makes up a protein, and that protein will leave the ribosome. Did I miss out on anything?
No, sounds good.
All right.
And then what?
And then what? And then that protein will go and become something, will become a piece of you, will be an enzyme, will be anything, these proteins. So that code that was nothing now is actually something tangible. So now we're just going to end with this quick 3-minute animation. Don't look at us because we're going to look awkward, but you want to look at the animation so you can see what transcription and translation is.
We never look awkward.
You never look awkward? That's true. It's just me looks awkward.
And here comes
the cell transcribes the genetic designs for each protein from strands of DNA to special molecules of RNA. An enzyme unwinds a segment of DNA, making room for its complementary RNA nucleotides.
So that's the
Both DNA and RNA nucleotides contain a sugar, phosphate, and base. But in RNA, the sugar is ribose, and one of the 4 bases is replaced by uracil. The exposed bases of the DNA strand serve as a model for the construction of messenger.
So you can see that that DNA that used to be a double helix, it flattened out and then it's separated, and this thing in the middle is the mRNA.
Or mRNA. On other strands, the cell produces transfer, or tRNA, and ribosomal, or rRNA. In their final form, these molecules play a vital role in protein synthesis. Translation begins with the binding of a messenger RNA to a small subunit of a ribosome, the cell's workbench for protein assembly. When properly positioned, the messenger RNA triggers the approach of a tRNA, which carries the first amino acid. The tRNA attaches only if its 3 nucleotides exactly match the first 3 coding nucleotides of the mRNA strand. A large ribosomal subunit now joins the group to form a functional ribosome, with 2 binding sites built from ribosomal proteins and rRNA. Soon, another tRNA arrives, which matches the next 3 nucleotides on the strand. With the help of the ribosome and some cellular energy, the neighboring amino acids bond together. The first amino acid then separates from its RNA taxi, leaving the ribosome in search of another, identical amino acid.
So I know it's kind of pixely, but what you have is, this is my mRNA chain, and then the tRNA, which we talked about, it came and it matched up. And then at the bottom of the tRNA, this green thing that you saw like magically connect together, that's the amino acid, and they start to connect together. And it's all happens inside a ribosome, so this little crusty thing is the ribosome. Do you have anything to add, Mr. Chu?
Peptide bond.
Peptide bond is how you connect these amino acids.
Now the ribosome moves along the mRNA strand. This exposes the next set of nucleotides, which match those of another tRNA. With each amino acid, the protein continues to elongate. As it grows, it folds into the 3-dimensional shape so crucial to its function. When the process is complete, the ribosomal fragments separate, free to join again later. With a legion of these molecules in operation, a single cell can produce hundreds of proteins every second.
So that's our quick, short video on transcription and translation, how you go from a DNA code to a protein. If you have any questions, you could ask me in class, or Mr. Jackson, or Mr. Shu, or Father, who's also here with us. So,
And Mr. McDowell.
And Mr. McDowell. So thanks for watching, and I hope you enjoyed this video.
God bless you all.
Video filmed in the 90s too.